So I'll be talking about the review article research that I did this summer, which is on the correlation between amyloidogenic mutated transthyretin variants and their localization as amyloid fibrils, which is in the disease called TTR amyloidosis. So what is hATTR? It's the hereditary form of the transthyretin or TTR amyloidosis, and is a progressive and eventual fatal disease that has a survival rate of approximately 3 to 15 years after disease onset, and it's characterized by the deposition of misfolded TTR proteins on multiple organs that eventually cause their dysfunction. And the misfolding is a result of genetic mutations in the gene, and different mutations are associated with different phenotypes. Currently, there are 120 identified TTR mutations, and the predominant phenotypes are familial amyloid polyneuropathy, which is FAP, and familial amyloid cardiomyopathy, known as FAC. So a general overview of the protein structure, it's composed of 4 identical monomers, as you can see in the different colors, the orange, yellow, green, and blue. And each of them have 127 amino acids. And they're also really high in beta-sheet content, which is the strips of rectangle that you can see. And TTR mutations are notated by the original amino acid name, its position, and then the new amino acid that replaces it. So for example, V30M, it's like valine, position 30, and it's switched to methionine. So the aim, since there are over 100 identified mutations that are known to cause this disease, as well as a diverse range of targeted organ localization, there must be some sort of correlation between the two. And thus, this paper aims to explore this correlation through synthesizing past research. And also because of its wide variation in phenotypes, hATTR is often misdiagnosed due to the similarity of its symptoms to other common diseases. And if there is indeed an important correlation, then this may provide insights into potential distinct features associated with certain phenotypes that may increase diagnostic accuracy. So methodology, initially I just gained like general understandings of the protein structure and the functions, and explored the 3D structure. I also researched about the most prevalent TTR variants, as well as the mechanisms of the aggregation process of TTR, as well as the structural changes that occur during that process. And the entire process is basically just identifying gaps and keywords in the existing research papers that I've read, and then further researching about those points. So my discussion was divided into 5 sections, and I'll be discussing some of the findings in these sections. So first, there are 2 types of amyloid fibrils that TTR aggregates into, and it's type A and type B, and their main differences are length and group arrangement. So some of the findings found is that in patients with the V30M mutation, which is a very common one, type A fibrils are associated with a late onset and cardiomyopathy, while type B fibrils are associated with an earlier onset and less myocardial involvement. But however, this is only restricted to this mutation and cannot be generalized currently. And a study also discovered that the C-terminal residues 51 to 27, which are present in both type A and type B fibrils, they're involved in the amyloid spine, which is an integral part of the aggregation process. And it's important to know that the majority of the most common TTR mutations are located at residues within this range. So second, it has been found that wild-type TTR, which is non-mutated TTR, possess a low inherent propensity to beta aggregation, and these single-point mutations simply enhance this potential, leading to earlier ages of onset and faster disease progression. Additionally, beta strands F, G, and H are found to be crucial to the aggregation process as a whole. Beta strand F increases efficiency, and amino acids in strand B enhances the potential, and beta strand H is a fibril forming segment. Furthermore, it has been found that there is an actual intermediate form that facilitates the entire amyloid formation process, and each of the intermediates are structurally similar with high beta sheet content as well. And it's currently unknown whether there is a primary sequence requirement for the forming of this intermediate. However, it's a crucial part of this process, and I think further research should be conducted regarding the structure. So for the popular mutated TTR variants, V30M, as I said, is one of the most common mutations, and it's associated with peripheral neuropathy, cardiomyopathy, upper limb onset polyneuropathy, and motor neuropathy. And the same can be said for many other mutations. There are mutations which are basically associated with a lot of different phenotypes, and there are currently no clear similarities found between these variants and the targeted systems or organs. However, for particular variants, such as TTR with the V122I mutation, it has been found to be kinetically unstable, as opposed to V30M and L55P, which are thermodynamically unstable. But these are also just limited to certain mutations currently. Some variants are also found to crystallize at different pH levels, some acidic and some basic. And again, no significant conclusions can be currently drawn for this information, as it's very limited to certain mutations. So for a specific type of hATTR, which is the leptomeningeal type, fibrils tend to localize to the leptomeninges, which are part of the protection layers of the brain. And these amyloid fibrils locate to the central nervous system. And since there is a blood-brain barrier between the CNS, which is central nervous system, and the rest of the body, it's a highly selective semi-permeable membrane. It is easier to examine the differences between TTR variants that localize to the central nervous system as opposed to other systems in the body. So in past studies, multiple experiments using different methods such as IP mass spectrometry and X-ray diffraction and many more have shown that TTR variants localizing to the CNS are largely unable to exist as stable tetramers in its fluid. And in comparison, variants that cause non-central nervous system amyloidosis are much more stable. Additionally, a study that separated the fluid in the central nervous system and blood into tetramer and monomer fractions found that mutated TTR variants associated with the central nervous system largely existed as monomers, which further confirmed the instability of these variants. And particularly, one of the variants, A25T, is found to dissociate into monomers about 1,200 times faster than non-mutated TTR. And also another mutation, D18G, is a monomer for more than 94% of the time. The correlation between stability of TTR and other traits. So the destabilization of the tetramer TTR is a key part of the formation of amyloid fibrils. And there are some correlations between stability found, but they also cannot be generalized to most TTR mutations. So the first is that increasing stability of these variants to acid denaturation in vitro is correlated with younger FAP ages of onset in vivo. For example, one of the most unstable FAP-associated variants, L55P, has a disease onset in the second decade of life, while a lot of other variants have an onset in the fifth decade, which is like 50 to 60 years old. And the second is that structurally, excessive proximity of the mutation site of mutations to nearby hydrophobic residues also have a destabilizing effect on the overall protein structure. So as we reach the concluding part of this discussion, I'd like to propose 3 areas of future research that can build on the current findings of this topic. So first of all, the residues 50 to 127 of the C terminus. 32 out of 77 of these residues actually form a structure that suggests the involvement of the C terminus in the amyloid spine, and further research into these 32 residues will prove extremely insightful. Secondly, because most of the phenotypes, including the predominant ones, involve organs outside the central nervous system, it is crucial to identify similarities and differences between these variants that cause system dysfunctions outside the central nervous system, as opposed to comparing central nervous system and others. And furthermore, differences in the primary sequence of the monomeric intermediates, if one exists, are especially important because they're so similar. And since it is an integral part of the intermediate, perhaps these differences will play a role in dictating the location of where these TTR variants will localize after they're aggregated. Thank you.